Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our next talk, Genetic Changes in Human Pluripotent Stem Cells, Implications for Basic Research and Regenerative Medicine. I'm incredibly pleased to welcome Professor Ivana Barbaric. Ivana completed her Doctors of Philosophy at Oxford in 2006 before moving directly into human pluripotent stem cell research, where she has established herself as a true expert in the field. Since 2014, she has been a group leader at the Centre for Stem Cell Biology in Sheffield. Ivana is a member of the Steering Committee of the International Stem Cell Initiative, General Secretary of the British Society for Cell and Gene Therapy, and lead for the genetic stability of stem cells within the UK Regenerative Medicine platform. A very warm welcome to you, Ivana. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And without further ado, I would now like to hand over to Ivana to begin her presentation.
Well, thank you very much for that kind introduction, and thank you also for the invitation to speak today. I'm delighted to tell you a bit about our recent work on the genetic changes in human pluripotent stem cells and the implications that we think this may hold both for basic research and for regenerative medicine. So why do we focus on these culture-acquired genetic changes in hPSCs? Well, I think we all know that pluripotent stem cells represent a really excellent source of cells for various cell therapy applications as well as for basic science. But the issue is really that majority of those applications require us to generate very large numbers of differentiated cells. Of course, this depends on the application, but if we take cell therapy of the heart as an example, there we may need to generate as many as 10 to the 9 cardiomyocytes. Okay, so we're talking about very large numbers of differentiated cells. And to produce such large numbers of differentiated cells, we may need to expand large numbers of undifferentiated stem cells and essentially keep them in culture for long periods of time. Now, what happens to stem cells as we grow them in culture is that they tend to acquire genetic changes, mutations, and then these culture-acquired genetic changes can have a range of adverse consequences on the differentiation efficiency of stem cells, but also on the maturity and functionality of differentiated cell types. And of course, from the cell therapy point of view, particularly concerning is the notion that these culture-acquired genetic changes in stem cells could confer tumorigenic properties onto differentiated cells upon transplantation into patients. And now what we know about the nature of culture-acquired genetic changes in hPSCs is, first of all, that they appear at all levels. In other words, we see everything from gains and losses of entire chromosomes down to single nucleotide variants. But when we look at how these genetic changes distribute across the genome and across multiple samples, then we start to see that they converge on particular genetic changes. So, for example, at the karyotype level, we tend to see the same karyotypic abnormalities time and time again. For example, gains of chromosome 1, 12, 17, 20, and X. Similarly, if we look at the single nucleotide variant level, here we tend to see recurrent point mutations in genes such as tumor suppressor TP53. And of course, these same karyotypic abnormalities, point mutations in P53, are also frequent in cancer cells. And it's this parallel between culture-acquired variants and cancerous cells that really raises concerns about the potential safety in cell therapy. Now, the non-randomness of these aberrations that we see, so the same genetic changes occurring time and time again, is essentially implying that these are the genetic changes that give selective advantage to our variant hPSCs. In other words, we may have mutations occurring in our cells all the time, but then it's those mutations, those genetic changes that confer selective growth advantage that will allow these variant cells to outcompete wild-type cells and dominate cultures. And as I said, this is the problem, or it's an issue, because at the end of the day, we want to use these cells for cell therapy, and we do not want genetically variant cells present. So to really understand this process and try to minimize variants from occurring, in my lab, we work on a series of topics to tackle this issue. And today I'm going to talk you through some of them, starting with really understanding how variants emerge in the first place, but then also understanding once they emerge, how is it that they, or which mechanisms they use to evade selective bottlenecks in culture systems so that they can dominate cultures. So if we think about the emergence of variant cells, obviously one way through which stem cells can acquire genetic changes is through mitotic errors. And this is why in my lab we study this process of mitosis. And we use assays such as this one where we we essentially do time-lapse microscopy on fluorescently labeled stem cells, and we monitor them as they progress through mitosis. So you can see from these types of videos that this process does not always go smoothly. And in fact, in about 15 to 20% of mitosis, we see an appearance of things like chromosome bridges or lagging chromosomes, or we see an appearance of a micronucleus. And all of these are indicative of issues during mitosis. One obvious explanation for such a high rate of mitotic errors could be high levels of genome damage. And indeed, we and others in the field have shown that pluripotent stem cells indeed suffer from high levels of genome damage. So in this example, we examined the same population of cells, so parental fibroblasts, reprogrammed to the pluripotent state and then differentiated again. And when we did assays that read out the level of genome damage, it's always this pluripotent state that has significantly higher levels of genome damage compared to the differentiated isogenic counterparts. So this is really important because it's giving us insights into the mechanisms through which stem cells may be acquiring genetic changes. And in this particular instance for genome damage, we went on to show that genome damage in these cells is caused by replication stress due to short G1 of these cells as they try to really progress quickly through their cell cycle in order to proliferate very fast. But what happens during such a fast cell cycle is that stem cells simply don't have enough time to prepare all the required metabolites for the ensuing DNA replication. And by knowing that, we were able to tweak or optimize culture conditions, and we added nucleosides to our growing cultures of stem cells and showed that by adding nucleosides, we can not only reduce genome damage, but also subsequently reduce mitotic errors. So as I said, the importance of this is really that by understanding the mechanisms that underpin mutations in the first place, we can optimize culture conditions and we can reduce the issues that may effectively lead to genetic instability of these cells. Having said all that, given the scale of hPSC expansion that we envisage would be required for cell therapy, or for other applications of hPSCs in basic science, it is possible, or in fact likely, that even a very, very low mutation rate could nonetheless give enough substrate for selection to take place. So even if we have very rare variants in these cultures, given the numbers of cells that we're expanding, that may be enough for those variants with advantageous traits to be selected for. And this is why in my lab, we also very firmly focus on this issue of selection. And today I'm going to talk to you about our findings about how the selection or the repertoire of variants that we eventually see in cultures is shaped by culture conditions in which we grow the cells. And by this, I mean not only the median matrix on which we grow the cells, but also it's determined by other properties, for example, cell density. Now, the revelation that culture conditions or median matrix influence how variants take over the cultures came from a recent large-scale retrospective analysis of a large number of karyotypes. So in this study, we collaborated with YCell, who perform karyotyping of hPSCs for not only Dell labs, but also as a service provider, they do this for a number of different labs. And so WiCell have a compendium of karyotypes collated from, say, 2009 to 2001, and this consisted of over 22,000 karyotypes. At the same time, in Sheffield, we also collated a much smaller number of karyotypes, but all of them came from our in-house data. And together, as you can see, we had over 23,000 karyotypes. So to the best of my knowledge, the largest compendium of hPSC karyotypes to date. Importantly, the majority of these karyotypes were also annotated with metadata. So we knew the culture conditions or the media and matrix in which karyotypes were sampled. And so really from analyzing this data, we were looking for potential association of chromosomal aberrations with culture conditions. If I just show you the overall data first from WiCell and then Sheffield, this very much on the heat plots, you can appreciate that this confirmed what we and others in the field showed before, and that is that genetic changes in hPSCs are not random, and that we see certain aberrations appearing in these abnormal karyotypes frequently, such as gains of 1 or 12, 17, 20, and X, for example. However, a big surprise for us came when we examined the karyotyping data set over time. So in this particular example, I'm showing you 6 most common karyotypic aberrations in the WiCell data set, which we plotted over time. So from this, you can see that, first of all, we don't have flat lines. So if we look back in 2009, over 60% of abnormal karyotypes in WiCell consisted trisomy 12. But if we fast forward to 2021, less than 20% of abnormal karyotypes contain this aberration of chromosome 12. On the other hand, aberrations such as gains of chromosome 20q, iso20q, or 1q were really not very prevalent back in 2009, but nowadays represent some of the most commonly detected aberrations. Data from our own in-house database in Sheffield is even more striking, although there's obviously less karyotypes sampled. But if we look in early 2000s, all of the Sheffield data sets had abnormal gains of chromosome 12 or 17. But then in recent years, we pretty much don't see gains of chromosome 17 anymore. And instead, over 50% of our abnormal karyotypes in Sheffield contain this gain of chromosome 1q. So what this really analysis told us is that there are changes in trends of recurrent genetic aberrations over time. And this is curious and has made us consider what is it that has changed over time in our cultures of pluripotent stem cells that could have caused these dramatic changes. And of course, one thing that's really changed in the field is the culture conditions. And I think we all remember that when we first started growing ES cells and then subsequently hPSCs, this was done on feeder layers in the presence of serum or knockout serum replacement. But of course, nowadays, the whole field has pretty much shifted to the use of feeder-free, KOSR-free conditions. And so because of this notion, we decided to analyze the karyotyping data set with respect to different culture conditions in which those karyotypes were sampled. So if we take as an example gains of chromosome 1q, in the entire WiCell data set, this aberration was present in about 9% of karyotypes. But then when we break this down according to different culture conditions, only 3% of only 3% of karyotypes contained aberration of chromosome 1q when cells were cultured or sampled in KOSR. In contrast to this, all of these other KOSR-free media, so E8, MTs, and Nutristem and StemFlex, contained much higher proportion of, or significantly higher proportion of gains of chromosome 1q. So really from this analysis, we concluded that there is a change in trends, and that these sudden or recent increase in gains of chromosome 1q is prevalent in feeder-free conditions, and in fact may have been caused by the field-wide shift from feeders to feeder-free conditions. Now, this is a hypothesis that fell out from our retrospective analysis, as I said, but of course it's something that we could validate in the lab. And we did this by taking 3 different lines with and without a gain of chromosome 1q. So we have isogenic cells H7, H9, MIF3, and their variant counterparts, so the same cells but now containing a gain of chromosome 1q. We then performed competition assays, and this really means spiking a small proportion of variant 1q cells into the wild-type culture and then monitoring the ratio of individual sublines in subsequent passages. 2 conditions we tested in competition experiments were KOSR-free condition. So for this, we used E8 vitronectin, and in parallel, we also used cells grown on feeders in the presence of KOSR. Strikingly, and entirely in line with our hypothesis, we noted that 1q cells, variant 1q cells, won or dominated the cultures only when they were grown in E8 vitronectin, but not when they were grown on feeders in the presence of KOSR. And this striking result was replicated across all 3 of our pairs of wild-type and 1q cells. So from this, we concluded that variant 1q cells indeed have context-dependent selective advantage. And of course, we were next then interested, what is really underpinning this selective advantage, or why is it that cells with a gain of 1q win only in these E8 vitronectin conditions, but not when they're grown on feeders in KOSR? So to address this question, we first postulated that it's possible that really wild-type and 1q cells have different levels of spontaneous differentiation when they are grown in different conditions. But when we checked the levels of markers of undifferentiated state, and this is just one example, SSCA3 shown on the slide, we noted no difference. So we concluded that this context-dependent selective advantage is not due to levels of spontaneous differentiation. On the other hand, we did see differences in proliferation ability of 1q and wild-type cells. So on feeders in KOSR, they behaved very similarly, but once cells were put on vitronectin in E8 medium, then we saw that variant 1q cells proliferated much faster. So we next looked a bit more deeper into this reason behind proliferative advantage of 1q cells. And for this, we performed these types of time-lapse experiments, which are very powerful because they allow us to track single cells as they try to reinitiate stem cell colonies. The lineage trees on the side are really made from the single cells in the videos, and the red crosses on the slide indicate cell death. So I think just from looking at the lineage trees, we can appreciate that there is way more cell death in these wild-type cultures compared to 1q. But another thing we noted by looking at and quantifying these lineage trees is that variant 1q cells, in fact, proliferate faster because they have a shorter cell cycle time. So they can go or get through the cell cycle much more rapidly. But equally, as I said, there's less apoptosis in these cultures. And in the end, because of the combination of these 2 factors, variants really are able to make much bigger colonies. And there are many more cells at the end of this video compared to the wild-type cultures. We also went on to identify at the molecular level the driver gene that is responsible for this phenotype of variant 1q cells. And for this, we focused on looking at the minimal region on chromosome 1q. So from our karyotyping data, we could identify the minimal region that's present in all of the variants with the gain of chromosome 1q. And in this region, we had a number of genes, but one really stood out, and that is MDM4. And it stood out because MDM4 is a known negative regulator of p53. We know that p53 is very important for human pluripotent stem cell biology. And we also know that p53 mutations themselves are recurrent genetic mutations in hPSC cultures. So it sort of made sense that amplifications of MDM4 could be another way in which cells could modulate p53 pathway and therefore gain selective advantage. Now, first of all, looking at the expression of MDM4, indeed, we see higher expression in cells with a gain of chromosome 1q, which is in line with them having an extra copy of this chromosome. And again, we see this across all 3 lines that we examined. Importantly, we also did functional experiments on MDM4, and specifically, I showed you from time-lapse experiments, I think you could appreciate that 1q variants have a much better cloning efficiency than wild-type cells. But when we knock down MDM4 in these 1q cells, then the cloning efficiency was dramatically reduced. Importantly, we also did converse experiments whereby we overexpressed MDM4 and then performed competition experiments again. So here, wild-type cells expressing MDM4 are mixed back with wild-type cells, and we look at their ratio in subsequent passages. And by doing these experiments, we could show that indeed, when MDM4 is overexpressed in wild-type cells, that entirely phenocopies the 1q phenotype. And we could also show that these wild-type cells with MDM4 overexpression have reduced apoptosis. So all of these experiments really strongly indicate that MDM4 indeed is the likely driver gene and gives anti-apoptotic resistance to our cells that have a gain of chromosome 1q. At that point, however, one question still remained unresolved, and that is, why is it that cells that have a gain of chromosome 1q, so have an amplification of MDM4, they give such a strong advantage to cells when they're grown in E8 on vitronectin? So why doesn't this work when we put cells into another culture condition, such as feeders with KOSR? And we found that the reason is following. So when we grow wild-type and 1q cells in E8 on vitronectin, then MDM4 expression is much higher in 1q cells. As I said, this is in line with these cells having an extra copy of chromosome 1. However, when we place the same cells in a different culture condition, feeder with KOSR, then we see that the MDM4 expression is now significantly dampened. And in fact, just the addition of KOSR into the E8 and vitronectin conditions pretty much depletes MDM4. And in line with this, we see that those cells now lose their proliferative selective advantage. So together, these experiments have, first of all, explained why selective advantage of 1q cells is effective or visible only in E8 and vitronectin, so feeder-free conditions, but not when cells are grown on feeders in KOSR. And importantly, by explaining this, we have explained why we now see the rising prevalence of abnormalities, abnormal karyotypes with a gain of chromosome 1q. As I said, this is something that's been seen in the field now with this shift to feeder-free cultures. So that was really an example of how culture conditions, or specifically BDI and matrix, can modulate which variants we get in our cultures. And this is really important because depending on which conditions we are using, we need to monitor a screen for our variants accordingly. So now that we know that gains of chromosome 1q are quite prevalent in feeder-free conditions, this is something that really we should be keeping an eye out for. But there are additional parameters in our cell culture practices that also influence the overtake of variants. And really, how we came to study this was really through an observation that some of the variants that we note in our cultures come to overtake wild-type cultures very, very rapidly, really way more rapidly than we could explain by knowing the cell cycle time or their rates of apoptosis. And that really made us consider the interactions between wild-type and variant cells and whether these play a role in how variants take over the cultures. So to examine these wild-type variant cell interactions, we performed again time-lapse experiments, but this time mixing wild-type cells, which in this case have nuclei labeled in red, and we mixed them 50-50, as I said, with variants that have the whole cell labeled with GFP. So when we monitor these cultures over several days, we first of all see that both populations are interacting, the cells are intermingling, both cell populations are proliferative. However, several days into this culture process, as the culture conditions are becoming more and more dense, then we see that variants are effectively starting to corral these wild-type cells and then effectively eliminate them from cultures. So this was a very striking behavior, as it told us that variants can be very aggressive and not only take over the cultures of wild-type cells, but actually deplete them or eliminate them from cultures. Now, this type of behavior is reminiscent of a cell competition phenomenon, which is quite well known and described in developmental and cancer cell biology. And we know from those studies that cells have different ways in which they can interact and affect each other's survival in culture. So, for example, they can do that through trophic interactions, competition for space, or the so-called direct fitness sensing that involves direct receptor-ligand kinds of interactions. And so, of course, we wanted to know which one of these plays a role in our cultures. So we went through these different options pretty much systematically. To look at trophic interactions, we performed the conditioned medium experiments as well as these transwell experiments where we mix different, we mix wild-type and variant cells in different combinations, but always making sure that they share the culture medium, but cannot come into direct cell-cell contact. But when we've done these experiments, we did not note this detrimental effect of variants onto wild-type cells. So we concluded that at least in our conditions, in our context, trophic interactions do not play a role in how wild-type and variant cells interact. So that left us with these two different possibilities. One is competition for space, and the other one direct fitness sensing. So here we postulated that if it is direct fitness sensing, then we should see death of only those wild-type cells that are in direct contact with variants. But if it's competition for space, then cell death of wild-type cells may happen several or maybe many cell diameters away from the nearest variant cell. So to tell these two possibilities apart, we turn to making these types of assays where we put wild-type cells and variants onto the fences so that when they meet, they come into contact at a pretty well-defined border. And as a control, we also had wild-type cells mixed with fluorescently labeled wild-type cells. So if I show you the control first, these cells come into contact and they maintain the border for about 48 hours or so. But in contrast, mixing wild-type cells with variant GFP cells, we can see that variants, again, are very aggressive. They are effectively pushing the wild type cells and invading their space, a bit like watching a rugby match, really. Maybe easier to appreciate from these still images. So the border that's created by the two cell populations is maintained in the wild type scenario. However, variants really invade this space. Importantly, when we look at where the apoptotic signal is located, it's not located only at the border, but it's also present many cell diameters away, telling us that the type of the interaction we are seeing here is mechanical or cell competition for space. We went on to characterize this at the molecular level, and really we showed that the key difference here is that when variant cells corral wild-type cells and push them into these really small spaces, then our wild-type cells start to sequester a key mechanosensor YAP into the cytoplasm, whereas the surrounding variant cells still keep YAP in the nucleus. And the outcome of this interaction is that the wild-type cells commit to apoptosis, whereas variants are still proliferating and surviving. But the importance of this finding was that for the first time, we were able to show that not only cell autonomous, but also these types of non-cell autonomous interactions play a role in how variants can take over cultures and enable variants to dominate and outcompete wild-type cells very, very rapidly. Also, from a practical point of view for cell manufacturing, this is pointing to the importance of cell density and really the fact that this is one of the key parameters that we need to think about and modulate when expanding cells for cell therapy and basic applications. And finally, just a quick slide to tell you about most recent work where we also showed that, in fact, it is not only during cell expansion that these variants are dominating cultures and exerting their effects, but actually these can also have implications for uses of cells in the differentiated state. So this is just a quick example to show you that when we, for example, take these panels of lines with and without a gain of chromosome 1q that I told you about, wild-type cells differentiate nicely into beating cardiomyocytes. But then when we, at the same time, try to differentiate cells with a gain of chromosome 1q, only in one of these examples did we get any beating. So that's in the H9 cells. We see a little bit of beating, but really the majority of culture did not differentiate. And that's actually seen from these percentages of cardiac troponin positive cells. So the implication of this is that, you know, if we unknowingly use variant cells in various applications, the efficiency of differentiation protocols can be massively reduced. And I'm not showing you the data here, but we also see that those cells that do manage to differentiate have very different phenotypes compared to their wild-type counterparts. And so I'll finish there, and just to summarize, we now know about recurrency of genetic changes in hPSCs. We're studying the mechanisms through which they arise and through which they dominate the cultures. But importantly, as we now see that these recurrent variants have really profound effects on differentiation efficiency and also phenotype of clinically relevant differentiated cells, I think this brings into focus the need to really bring about strategies to minimize these variants from occurring in culture, but also the need to really think more deeply about which variants should we really care about and be careful in terms of using them in cell therapy and other applications. And with that, I would just like to acknowledge all of my lab members who contributed to various aspects of this work, and also all of our collaborators, and thank you for your attention.
Thank you so much, Ivana, for the fascinating talk. It was truly excellent. As with all the other talks in the event, we're likely going to have lots of questions come in from the audience. And I can see that we have received some questions already, so I'll move straight onto the Q&A now. So your first question is, apart from karyotypic abnormalities, are there any point mutations that are recurrently gained in hPSCs?
Indeed, karyotypic abnormalities were historically the first ones to be identified. And this is partly due to the fact that when we started this research, or the first observation of recurrent genetic changes made exactly 20 years ago in 2004, back then the main techniques that were used for characterizing cells were things like karyotyping by G-banding. And so those were the most likely to pick up on karyotypic abnormalities. But of course, over time, there's more and more use of next generation sequencing technologies. And as these are becoming more prevalent, we are also starting to gain more understanding and insight into point mutations arising in these cells. And a particular example that I highlighted during my talk, and particularly concerning one, I would say, are mutations in P53, because they're the same dominant negative mutations that are often seen in human cancers. But apart from P53, there are also several other cancer-related genes that are often, well, not often, but have been seen in as mutated in hPSCs. And one that also stands out is BCOR, which has been reported as rather frequent too.
Thank you so much, Ivana. That's fantastic. I am aware of the time, so we have enough time for one final question today, which is, how often should hPSC cultures be screened for the presence of genetic changes?
Okay, so that's a very pertinent but also difficult question to answer, because we really don't know exactly when these occur and how quickly they can take over the cultures. But based on what we know of the recurrent variants, we would recommend monitoring the cultures maybe every 5 to 10 passages. And in fact, last year, the International Stem Society for Stem Cell Research, ISSCR, has published some guidelines or standards for the use of human stem cells in research. And they, in those guidelines, stipulate that, first of all, banks of, where possible, of cell lines should be made, and that the cell lines should be used within 5 to 10 passages from the point of banking or defrosting, because just by minimizing the time in culture, we minimize the possibility of occurrence of these variants. And the monitoring for changes should be done in those timelines, but also whenever scientists notice something different about their cells, if they started to grow abnormally. And finally, if there is a particular bottleneck that the cells went through, if you notice that you've done something that's killed majority of your cells, then it's probably a good time to check your cultures afterwards.
Thank you so much, Ivana. Unfortunately, that is all that we have time for today. As mentioned, any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thank you once again, Ivana. It's been an absolute pleasure to have you present with us. And to everyone in the audience, we will be back soon with the next talk.